Justin?
Brilliant. All right, thanks for the introduction, Incarnita. And thanks to our speakers in the first half. That was a really interesting introduction to the biology behind specificity. And now in this second section, we'll take a look at how that's modeled computationally. And we're fortunate to start off with Victor Greiff, Associate Professor at the University of Oslo and Director of Computational Immunology at IMPRINT. Please go ahead and take it away, Victor.
Yes, Justin, thank you very much. Just need to start sharing. Yes, I will talk about mostly from the viewpoint of antibody specificity. And I have a slightly grandiose title, so please excuse this title, but I didn't really know what else to choose. And I will also talk from an experimental point of view. So I will cover a bit of computation, a bit of experimental, a bit sprinkled in with our own ongoing work. But I I largely followed Encanita's lead, who told us to be a bit provocative and ask questions. So that's what I'm going to do. Um, So to just frame the problem of binding prediction, and here I talk about prediction of antibody binding, but it's largely also similar for TCR binding, is that we have this overwhelming diversity of different antibodies and this very low overlap of immunoreceptors across different individuals. And each antibody on the single chain is about 110 amino acids long, and the length of the antibody binding region depends on the structure, of course, but it's relatively short. And that means, since to the shortness, and since we know that we can find an antibody against almost any given uh, um, antigen, there must be higher order sequence dependencies that mediate antibody specificity. And what we mean by these higher order dependencies is basically these motifs that mediate paratope-epitope interaction. And as we already heard in the previous talks, specifically David mentioned it, that we really need to know when we talk about specificity, and I will go more in detail also on the next slides, we are also we need to talk about context in terms of what we are looking at, and we cannot just define specificity as affinity, but as also selectivity with respect to a landscape of targets. But I will go more in detail in the next slides on that. Briefly, just to set the stage, is that at least for antibody binding, most of antibody-antigen interaction, at least as seen from a structure point of view, is located in the CDR, in the CDRs 1, 2, and 3. So in the study that we published in 2021, we saw, and there we looked at, this was pre-COVID structures, and these were about 800 to 900 crystal structures. And we looked at the location of the interacting amino acids on both the heavy and the light chain. And you see that most of the interacting residues locate to the CDRH1, 2, and 3, and less so to the framework region. So the framework regions on the heavy chain, at least, only 10% approximately, and the CDRs 90%. And I think it's similar it's similar on the light chain as well, but the proportion of where the majority lies changes with respect to H1, L1, and so on and so forth. So that's just a bit of biological background. And I think it would be nice to redo this study now with more structures available. Just so we're on the same page of how we define affinity, that's actually taken from my PhD thesis. So I don't think I have to go in detail, but it basically defines the strength of ligand-protein interaction. And here is the formula for KD, and KD can also be zoomed in as the on and the off rate, which quantifies the rates of at which a free antibody and a free antigen combine to form a binary complex. That's the on rate, and then the off rate is the dissociation rate. Um, um, and then, so this was affinity, and this is one antibody ligand pair. And how do we define now specificity? And also, this is from my PhD thesis, and I have the citations here. One is from Greenspan, and one is from Van Regenmortel, who I think define it really well, and it's exactly in line with what David said before in terms of the lamppost thing. Um, if we define affinity as delta G, specificity can be defined as delta delta G that characterizes two or more receptor ligand interaction. And thus, the definition of specificity depends on the frame of the reference. The local environment of the studied species has to be given, as well as the chemical species to which the studied species is compared to. So in normal English, this means basically you need to know that you are working under a lamppost, and you need to know the light that is um, illuminated by your lamp. If not, you cannot really say whether your antibody is specific or not, or TCR. And saying it a bit more smoother maybe is you need to, when people talk about specificity, they need to provide the reference to which specificity has been quantified. And I don't have to go over this, but it's basically similarly also been defined by Van Hagen model, who then
Cómo están juntos, pero a veces que le pongo aquí, mira, TP, pero me aparecen los.
who said that specificity is basically the discrimination potential of an immune receptor. Here he mentioned he talks about the antibodies, but same for TCR. And then David also previously defined different levels of cross-reactivity. And I think this is one of the diseases of our field, that we don't have a common agreed upon definition of what we mean when we say cross-reactivity, polyreactivity. Everybody has their own definition. There's not one dictionary where we can just go and look up what has been agreed upon for a given word. So I think this is something that we should resolve as soon as possible. And I don't really care about the words. I more care about how those terms are defined. And I think we as a field need to do better in defining these very important concepts, because only when you have the definition of a concept, you can perform experiments that elaborate on this concept. So if here again, happy to discuss this afterwards, but I think we all know that antibodies can be cross-reactive, which means that a given antibody can react to many different targets with similar affinity, and we could subdivide it as by Robert et al. In a 2018 review, which I highly recommend reading. Very nice, very clear, where they define several subcategories, which are again named by different people differently, but you can find this cross-reactivity in binding to unrelated and diverse antigens, muted variants, or binding to closely related antigens where the epitope is conserved. So there are many different degrees of cross-reactivity, and once we have a good definition of how we define these concepts, I think it will be much more productive to then also perform experiments to go in deeper and understand them mechanistically. Just also highlighting a personal, or not a personal, just something I think that is very underexplored is the concept of serum antibody specificity. So the concept of how the polyclonal mixture of antibodies in the serum binds as a whole, as opposed to monoclonal antibody specificity. And here Talmage in his very famous specificity paper in Science in the 70s or 50s said that in a mixture of large number of different globulin molecules, the dominant reactivity will be that common to the largest number of molecules present. And I don't know how he could say that with that certainty. But this is something that I saw in my PhD work as well, that where we did mathematical modeling of serum antibody mixtures and where we found that the ensemble specificity of the serum really depends on the the frequency distribution of serum antibodies, and this is something that I would love to go on deeper in the next years, given that we have an upgrade in technologies to really study this at a one-to-one level. But this was just a side remark going forward. And then just broadening it again, okay, now we know that there is cross-reactivity, but to what extent can we actually predict antibody binding? This is a question that as computational people we get a lot. And I, for once, have hope, not only because there are a lot of data being generated, but also the biology gives us hope because we know with the current data that has been generated, we know that actually now the antibody sequence space is much more restricted and constrained than we previously thought. So we now know, thanks to the work of Walczak and Mora, that and others, that VDJ recombination statistics are largely predictable. Our work has shown some evidence for a common vocabulary of antibody interaction motifs that are shared across very different antibody-antigen structures. And also, all of us know, both in the T cell and the B cell field, about public clones, right? So you immunize different individuals with the same antigen, and these individuals will either form or have already or will evolve to some extent shared immune responses. So there is some constraints and restrictions in our immune receptor space, and I think, especially for the computational field in the next years, it will be very important to define those constraints and use them as priors to increase predictability of binding, for example. Now we come to what I had warned you already, me just asking questions and not having answers. So I structured this into experimental and computational questions. So on the experimental side, of course, there are many questions that had been raised by David and also Antonio in terms of biology. Here I just raised basically technological questions. So how can we actually generate the data that we need for prediction? And we can ask this for antibodies on the BCR level, so BCR antibody-antigen interaction, and how can we measure this at many to many, so many antibodies versus many antigens, and same for serum antibody binding, which is more a proteomics problem. So one is a sequencing problem and one is a proteomics problem. And not going into detail, but also just showing here again that there are some technologies that, if invested in more heavily and performed at higher throughput than currently at least publicly available, I think we would be in very good shape to build great training data sets for antibody-antigen binding prediction. So there are technologies now available for library analysis, meaning basically you can screen many antibodies versus many antigens. So this is one technology, for example, this is which you can buy from Alpha Bio. So you can buy their datasets and they can make datasets for you. We can generate many-to-one datasets, which are affinity annotated as well, at the order of hundreds of thousands versus one antigen. On the screening the natural repertoire, there are technologies such as LibraSeq by the Georgiev lab, who also now got a $30 million grant from ARPA-H to really build an atlas of antigen-specific antibody sequencing as sequences. So this will be great to see published at some point. And then from a structure point of view, there is a lot of work by Andrew Ward's lab on trying to define the polyclonal antibody response structurally and using also proteomics. And a few other labs are working on this as well. And then, of course, there's a lot of repertoire screening work with peptide arrays or FIPSeq and so on. But here, this is a great technology for looking at a serum antibody binding at a broad scale. Unfortunately, these technologies, as of yet, do not allow for knowing which antibody has bound to which epitope or protein. Yes, so there is hope, but I think the summary of that is we need more investment in experimental technologies to screen specificity of immune receptors. And I think that goes both for TCR and BCR. Just to show you really, and this is really thanks to the advances in synthetic, so phage display, yeast display, and mammalian display. So here is work from Sai's lab and our lab where we screened, where we mutated trastuzumab, which is a very known monoclonal against HER2, which is a breast cancer antigen, and we mutated just the CDRH3. And what we found is that tens of thousands to hundreds of thousands of sequence dissimilar variants that can still bind the paternal target epitope. So really showing you how immense just the diversity space to one target epitope is. And which brings me, so if that is true, then this brings up a lot of questions. How can very similar antibodies bind to entirely different antigens and the other way around? How many mutations are needed to convert an antibody from one epitope to another epitope? The very classic question, are there holes in the repertoire? What's the density distribution of antigen binding across the antigen space? And to what extent are BDJ recombination probabilities in germline gene alleles the subject of pathogen-specific evolution? Many questions, all of which of course I cannot cover and which will require many lifetimes to cover. I would just go in very briefly into a few questions that we are trying to tackle right now, which is this one question: how can very different antibodies bind to identical epitopes? And just very briefly saying again, in order to understand this many-to-many binding of immune receptor antigen binding, we really need many-to-many binding data. And this is true for both antibody and antigen. I think we are a bit further along on the antibody side, but I think the T-cell field is also catching up there. So I really want to say, and both for T cell and for B cell, we need to understand the effect of small mutations on antibody function in order to predict binding and specificity. So what very briefly, what we are trying to do in the lab is we did a very in-depth experiment looking at 5 different variants of trastuzumab, which differ in at least 5 amino acids in the CDRH3 um, with respect to the wild type. And we looked at them with SPR, so we made sure that they are still high affinity, and we looked at them with HDXMS and with QIUM, thanks to Andrew Ward's lab. And we and then we did molecular dynamics simulations and as well as docking to really understand how it can be that very dissimilar antibodies can still bind the same epitope. And we found basically that paratope-epitope binding is a sum of weak effects, that there are multiple epitope sites that can support binding, that binding is usually supported by a summation of weak and diverse paratope interactions, and of course, which we have known for many decades, the flexibility of CDR loops. Um, and importantly, what we showed in this work that is pre-published in this workshop, but we're working on the peer-reviewed submission, so it should be on bioRxiv relatively soon, is that we could show that based on all these data, as well as our computational simulations, we could show that we can basically recapitulate what we saw on the experimental data also with our computational modeling, which we think is a prerequisite to going then to much larger data sets. So not just 6 antibodies, but in the future we are now looking at hundreds of thousands of antibodies and trying to understand what makes a binder and a non-binder a non-binder. I would also like to say, and I don't have to say much about that, that protein dynamics are central. Most of us are not looking into that, but in order to understand binding, we need to understand binding in space and in space, but also time. And for example, previous work by other groups has shown that that the paratope that is the binding competent conformation already exists in the non-bound antibody, for example, if you look at it from a molecular dynamics perspective. So there's a lot of things, I think, in merging sequence and structure analysis that can be explored in the future. And since molecular dynamics simulations are very expensive, there's a lot of work being done also in the machine learning field to speed up and reduce computational resources. Then I would just like to highlight one recent paper that really amazed me, which is work which was published just last week in Immunity. We are not involved at all, which is by the Duran-Besemann lab, where they showed that in mice, that in monoclonal mice that have only that have only one antibody kind against antigen X, and you immunize them with antigen Y, after a few immunizations, these mice can make antibodies that are highly specific against antigen Y, which goes, at least to my understanding of the textbook German Center reaction, against the dogma that you need to have at least a baseline reactivity against antigen in question, this one, to start the German center reaction. So they basically showed that there are clonal trees where the paternal antibody is antigen X reactive, and then the downstream antibodies in the tree are antigen Y reactive and not being cross-reactive anymore to the X antigen. So I found this really amazing. So I think we don't know so many things about antibody evolution. That was just a side comment. To my last point, I think we also need to better understand, no, it's two more points. We need to better understand the epistatic landscape of antibody binding. There is work in the protein evolution field which claims that just by knowing single and double mutants, you can predict all higher or up to 96% of all higher order mutants. I would be very interested in if that is also true for the antibody case. And then the last point that we are a bit interested in now is the density distribution of antigen binding, which we don't have so much data for yet, but we're trying to develop computational frameworks to understand this. And that goes towards building or at least a computational framework for building an atlas for immune receptor binding. So we all know Human Cell Atlas, great initiative, but where is the atlas of antigen-specific immune receptors? And I think Brian will talk in the next talk a bit about this, and we should probably talk more also offline. So what we are trying to do is trying to use protein language models for that, and we are especially interested into the claim which has been made by either explicitly or implicitly by antibody protein language model papers that protein language models group antibodies of similar function, for example, binding, affinity together. But first we looked at in general the overall shape of the immune receptor space, and what we find is that if we just take a common antibody protein language model and immune receptors from iReceptor, we see already that the shape of the final cluster is already predetermined by just very few sequences. And I think you can even go down to 1K sequences and you still find that the overall shape of the immune receptor space is conserved, which goes against to my first few slides, that there is some underlying constraints in the shape of the immune receptor space. And yeah, we controlled for random shuffling and so on. I can just move on. And then the question that I raised, that we were really interested in, to what extent can we use protein language models to group immune receptors of similar specificity together? And for that, we defined a score which we call the vicinity score. So the higher the score, the more similar or the more identical binding labels we have in the vicinity of a given immune receptor. And we turned again to our trastuzumab dataset that I already talked about before, where we have 100,000 binders and non-binders against HER2. And we found that, at least in our hands, most of the time, protein language models are not better in grouping similar labels together than a simple edit distance approaches. But then when we went to other deep mutational scanning datasets where they not only mutated in the CDRH3 but outside the CDR3, then we saw some advantage of protein language models. And we looked at many different encoding and pooling types and also layer depth, and we're working now also on a publication on that where we will go much deeper into all of these kind of findings. With that, yeah, and just to wrap up on this protein language model part, we're trying to really come up with quantitative metrics to compare immune receptors in high-dimensional embedding spaces. I want to highlight that we need, for really robust prediction, we need competitions, some of which are led by our community. And as we laid out in a recent review, in order to solve this problem of binding prediction, we need to work together in networks such as ours. And with that, I would like to thank everyone, and sorry for going a bit over time.
All right, thanks very much, Victor. So quickly to get your questions in, as we only have a few minutes here, I see Simon has his hand up, so we'll start with you, Simon.
On one of your earlier slides, you had the antibody binding sites mapped to the CDR regions, and the CDR3 has fit nice to the binding region. But do you think that CDR1 and CDR2 are not very defined properly? So if you take the usual definition and look at your data, the definition is too short. And if we use our software that annotates stuff to the CDR regions, and we look at CDR mutations and binding inside CDR regions, if our actual binding sites are outside the CDR regions, maybe we have to overthink what the definition of CDR1 or 2 for future analysis.
Yes, I fully agree. So I think I'm not sure what, I think we use the structure-based definition of antibodies and or of CDRs and frameworks, but what we saw is what you said. So at the corners of it, at the edges of the CDRs there, which are frameworks, there are also a binding involved amino acids. So yeah, I fully agree. Yeah.
Jamie.
Hey, Victor, good to see you giving such a great talk. Thank you so much. Yes, so I wanted to ask you, back in the day, people were taking the Chothia structures essentially that are determined by, the genetically predetermined CDRs and looking at their combinations, which will give you, in not all, but some cases, and of course, this was done a long time ago in the 90s, but gave these topologies. And you could see where they were shared, for instance, in some combinations, where you would get like a ridge, that a peptide could fit into, as opposed to, other kinds of topologies. And I was wondering if in your modeling and your, AI work, if you had taken things out of PDB and combined them with looking at the VD, the CDR combinations essentially, or the genetics, either way you want to look at it. Not so much focusing on CDRs necessarily, to see if you can look at shared topologies, which I think have a lot to do with, and also, certain residues sticking out, especially if they're hydrophobic or charged,
Yeah.
that kind of thing, because those are the interactions that mostly drive, salt bridges,
Yeah.
CDRs, and all that. So where you said earlier that binding is fuzzy and you don't see critical binding residues, I don't think that's exactly true. I hope,
Oh, I don't say
because
that. No,
there's a
no.
huge literature on the fact that there are.
No, no, I don't say that. I don't think I ever said that. Um, I, I say that a given antibody can bind many different antigens. And of course, one antibody and one antigen, there are specific residues that define the binding energy. So I think we fully agree on that.
Okay, that's good. But
What we
also
what we see
the
is
affinities. I mean, when
but
you say can bind a lot of things, well, yeah, they can bind a lot of things weakly, but maybe only certain things strongly. I mean,
exactly. No,
yeah.
yes, yes.
So I mean, that statement again, it's a little
Uh,
disturbing to me because
Uh, I think it is not disturbing because in the old work, they didn't test a given antibody against many different antigens. And if you give a monoclonal antibody on a peptide array, you will find many antigens that will light up with very high affinity against completely different structures. And maybe they share common binding motifs. And this is what we have to find out using structural tools, etc. Yeah,
Well, and therefore if they have common binding motifs, then they have to have something in common.
exactly. And this is what we need to find out. I fully agree with you.
Yeah, yeah. So
Right.
anyway, I was just thinking that if you can look at topology, I think that may be
Fully
very,
agree. That's what we are on.
yeah.
Yes, I fully agree. Yes.
So very quickly, Kenneth, for the last question.
Hi, Victor. That was great. I was just wondering if in your work you had found anything interesting with regards to how the light chain relative to the heavy chain affects affinity and specificity.
Yeah, we are largely ignoring the light chain in our work. So at least in the deep mutational scanning work that we showed with trastuzumab, we keep the light chain always constant. So we don't change the light chain. Although even especially for trastuzumab HER2, it is known from the structure that the light chain is involved in binding. So for now, we are ignoring it just for sanity and data size and complexity. But of course, in the future, you would like to look at both at the same time. Yeah. Yeah.
Thank you.
Fantastic. Thanks again, Victor, for that very provocative talk.